Welcome to the Ricci Flow Nutrition Podcast. My name is Cameron Borg. On this episode, I had the pleasure of speaking with Stephanie Seneff. Stephanie is a world-leading expert on the herbicide glyphosate and its host of detrimental effects on plants, animals, and microbes. Her publications and book, Toxic Legacy, have laid the foundation for understanding the implications of widespread use of this weed killer, particularly as it pertains to developmental disorders and metabolic disorders. She has also contributed greatly in the fields of sulphur metabolism, statin drug side effects, heavy metal toxicity, and more. Stephanie was the first guest I had on the podcast back in 2021, so it's a real pleasure to be able to speak with her again and expand on what she's been working on. In this conversation, we center our discussion around deuterium, the heavy isotope of hydrogen. Having recently spoken with Gabor Chomalier, who is the father of deuterium science in biology, this conversation follows very naturally. I sincerely appreciate Stephanie's work, as she has put her reputation on the line many times to pursue interesting and fruitful ideas that often clash with mainstream dogma. I think many of her ideas will be proven eventually as they follow an elegant logic. So with all this being said, I hope you enjoy our conversation. Stephanie, thank you so much for coming back on the podcast to speak with me. You seem to have decided to look into some of the most difficult topics and broad-ranging topics. Last time we spoke about your book that covers the topic of glyphosate quite well, quite in depth, and you've become quite passionate about this deuterium story that I've started to get into very recently. So, how did you first become? How were you first introduced to this story, and what did you make of it initially?
Yeah, well, it kind of has to go all the way back to 2007 when I got interested in autism. So I was seeing the rates go up. And for 5 years, I studied looking at all possible different kinds of exposures they might be having and striking out. 2012 was when I heard Professor Don Huber speak on glyphosate. I have a book here, Toxic Legacy. This was published in 2021, so almost a decade later. Having done a lot of research on glyphosate, Toxic Legacy, how the weed killer glyphosate is destroying our health and the environment. So I learned how glyphosate, first of all, glyphosate is causing an increase in a huge list of diseases that are chronic conditions that people are experiencing today in the United States. I believe glyphosate is the primary cause of these illnesses, of course, autism especially. And I learned about glyphosate's unique mechanism of toxicity, which I believe is true, that it's substituting for glycine at critical points in certain proteins and causing them to misbehave. And I had even identified which proteins. I talked about a glyphosate susceptibility motif and which proteins would be most likely to be severely affected. And then to show that literature was showing, actual studies on glyphosate were showing that it was suppressing these enzymes that I would predict it would suppress if my theory was correct. So the dots are connecting extremely well. And all the features of autism, the autistic kids have a lot of issues besides their brain dysfunction that can also be explained through the idea that glyphosate is messing up these enzymes by getting into them by mistake in place of the coding amino acid glycine. Really critical point that I make repeatedly. Yep. So I was in place with all of this information when I actually had published a paper together with Dr. Greg Nye. He and I have collaborated on a number of papers. We've really enjoyed working together. And he and I are both sort of experts on sulfur. We both had independently gotten into sulfur and the sulfur system in the body and how that's disrupted in certain diseases and how glyphosate disrupts it. So we've had a great time collaborating. We published a paper in the journal Water on sulphate and gelled water and cobalamin deficiency, and then linking it to glyphosate. We had this paper, talked a lot about metabolism and how glyphosate impacts it and how it can cause these different conditions and eventually affecting the brain. And Laszlo Boros, Professor Laszlo Boros, read that paper, was impressed with it, reached out to me. So it was just a cold email that I got from him saying, hey, great paper. And by the way, deuterium is basically what he said. Wow. Do you know about deuterium? And I said, no, I don't know about deuterium. Tell me more. And so he and I have struck up a friendship, and he's been awesome. He's definitely helped me to learn about deuterium and its roles in the body and how it's managed by the body. And it didn't take me a second to realize that the enzymes that glyphosate disrupts are often the same enzymes that are managing deuterium. In other words, if glyphosate is disrupting those enzymes, deuterium is going to be in trouble. So I became very excited about deuterium. That was in December of 2019, right before COVID hit. And I got into really digging into the research on deuterium. And then, of course, COVID hit. And then I got interested in COVID and linking COVID actually to deuterium deficiency. I believe that, or deuterium mismanagement, I should say.
Yep.
I believe that the people who are most susceptible to severe COVID are the same people who are being poisoned by glyphosate. And glyphosate is disrupting a lot of proteins in their body that would have protected them from COVID. And because of those disruptions, deuterium becomes problematic in the mitochondria. The mitochondria become dysfunctional in the immune cells, and then the immune cells can't fight off the virus. And then the really fascinating thing is what goes on in the lungs to try to fix the problem in response to the viral infection. There's a massive response it's a fascinating response that takes place in the lungs. It causes them to fill up with fluid, and all these things happen. I can talk more about that later, but basically that's an attempt, I think, to rescue the mitochondria of the immune cells with respect to the deuterium toxicity problem so that they can fight off the virus. So that was a long-winded answer to your question, but I am super excited about deuterium, and that should be very clear to you.
Yeah, well, I was just about to say, it must have been very exciting for you joining these dots together and having these epiphany moments where you're realizing that the things you're interested in are quite intimately connected,
exactly.
or at least seem to be in a very direct way. I think it would be worth sort of recapping the glyphosate issue that we spoke about last time. Maybe I'll just give a small rundown and you can tell me where I've gone wrong. So,
okay.
Glyphosate is this unusual molecule that is almost like a glycine, so an amino acid, but it's got some extra stuff on it. So it's a little bit larger. Not only is it a metal chelator, it's also an antibiotic. And it appears to be able to, in certain places like collagen, where it's very long and stretchy, it's able to jump in instead of glycine and interrupt the way that the proteins are folding.
That's right.
We don't know, I guess we don't know what the whole implications of this this glycine substitution might be. But, our extracellular matrix, all the stuff between all of our cells is made up of a lot of glycine-containing proteins. So
that's right.
We don't know what the implications of this might be if it turns out to be true, which I think it's, the writing's on the wall. It'd be very difficult to argue against this glycine substitution, hypothesis, if you will. So basically, we've got this water-soluble toxin in the environment. We can't get rid of it because not only do we keep spraying it, it doesn't break down very easily, and it appears to embed itself within us. So we're facing this big problem where we're not really sure what to do about this, and we're not sure to the full extent of how this is affecting us. And it looks like not many people are willing to look into the problem either, because it's a big deal if you're saying, hey, this is actually hurting people really badly. Is that a decent brief summary?
That's very, very good. Yes. And of course, the thing is the farmers at this point seem to be convinced that they can't live without glyphosate, and they're telling the government we can't do without it, which is really, really sad to say. We're going to be willing to poison our children, all of us, but especially the kids. It really breaks my heart to see what it's doing to the children. And we can't stop doing this because it's so essential for food production. That just cannot be true, but that's what they're saying. And of course, the industry fights really hard and claims that the chemical is perfectly safe. But there are a lot of papers coming out in the last few years that are very clearly showing evidence of damage to all kinds of different situations. So I'm excited about the recent. I think for the longest time, people didn't bother to study glyphosate because it's safe. It's sort of boring to study something that's not going to show any trouble. So they were convinced that it was safe, and therefore they wouldn't spend their money trying to study it. But now that people are becoming aware that it is toxic, more and more researchers are finding it necessary to actually do the studies and to show what it's doing. And they're finding a lot of evidence that I would say that I'm right, in terms of what's happening.
Yeah, and I recently spoke to Kerry Gillam on the podcast as well, and we spoke about the the class action lawsuit in America, that I believe 15 or 16 billion dollar class action.
Right.
That wouldn't have got hold if there wasn't at least some truth to this idea that it's
Exactly.
causing non-Hodgkin's lymphoma.
Right.
And that's just one type of cancer. This is a very narrow slice to what I think is really going on. So,
Yes.
and they wouldn't have conceded if they knew that it was safe. So
Right.
it seems to me like the writing's on the wall. How does this directly connect to this deuterium story where, we're in an environment where basically it's very easy to accumulate more deuterium than we would have in our evolutionary past, more than might be good for our general health. How do these stories intersect?
Yeah, so it's complicated, of course, and I'll try to make it as simple as possible. But let me start with what deuterium is, because I suspect a lot of your audience doesn't know. So if people know about hydrogen, it's the smallest atom. On the periodic table, it's the guy up on the left-hand corner. Only one proton, only one electron, really tiny, and incredibly prevalent. It's by far the most common atom in our body, and it's incredibly involved in all these redox reactions that are part of metabolism. It plays a role in just about every reaction that takes place, and often an essential role. So hydrogen is very, very important. Of course, it's water, H2O, two hydrogens and an oxygen. That's water, and so it's essential, of course, and it's a very important molecule. But so deuterium is heavy hydrogen. It's also an atom. It's a natural atom. It's found in nature. It's found in seawater, for example, at 155 parts per million. So it's a rare isotope of hydrogen, same as carbon-14. It's an isotope of carbon. And the difference between hydrogen and deuterium is that deuterium has a neutron as well as a proton and an electron. It has a neutron. And that neutron is about the same weight as the proton, so that makes it twice as heavy as hydrogen. And because hydrogen's weight is just one, adding one more is doubling, and that makes a huge difference in how it behaves. So it's interesting, and it's sprinkled throughout. Everywhere there could be a hydrogen, there could be a deuterium substitution, anywhere in any molecule. And you don't know where they are, but they're randomly distributed throughout all the molecules that are in our body. But our metabolism knows about it and pays huge attention to it. That's what's super, super interesting. And the game that the metabolism plays is to try to make sure that there's as little deuterium as possible in the mitochondria. The mitochondria hate deuterium, and that's because it messes up the ATPase pumps. These are the proteins, these are enzymes that actually make use of hydrogen motive force. So the hydrogen gets pumped into the intramembrane space of the mitochondria, and so there's excess hydrogen in there, and then they try to get back out, and they come out through the ATPase pumps, and the proton motive force of those protons spilling out is what gives the energy to make ATP. So that's the whole process of metabolizing, for example, sugars and turning them into carbon dioxide and water, and at the same time producing ATP. And ATP is the energy currency of the cell, so very, very important. Mitochondria are essential for the cell's well-being. And the mitochondria, if they've got a lot of deuterium atoms knocking through that system, they're going to break the ATPase pumps. They're going to jam them. They're going to break them. It's like sugar in the gas tank. I like to think of it as sugar in the gas tank. They don't work well. And that's why the whole mechanism of metabolism is centered on delivering as little deuterium as possible.
And this has
those
been
protons
shown,
are pushed in.
This has been shown, hasn't it, that the deuterium breaks the ATPase rotor?
Yes. Yeah, there's papers on that. And it's quite fascinating. And that's the critical. And then when you look at metabolism in a new light, once you realize that, then all of a sudden things make sense. And I had been fascinated by the fact that, for example, looking at cholesterol metabolism, and you find that there's this escort protein that brings cholesterol into the mitochondria. And then there are the CYP enzymes in the mitochondria that actually metabolize the cholesterol, putting hydroxyl in there, modifying the cholesterol to make hydroxycholesterol. And then the cholesterol is taken back out of the mitochondria, and then the sulfate's added to it. So I'm like, why? And you have to put the enzyme in there too. So you've got to put all these things into the mitochondria to let the reaction take place there. And then to take it back out. And it's like, why does it have to be in the mitochondria? Why do some reactions have to be there and other reactions have to be in the cytoplasm? And I got interested in that before I knew about deuterium.
Right.
But I couldn't figure out why it would do that. Why would it go through such effort, to to make sure that this thing is taking place in the mitochondria. Once you realize deuterium, it makes complete sense because all these enzymes, and I'm always looking, where is this enzyme acting? Where is it working? If it's in the mitochondria, it's going to be helping to make deuterium depleted water, almost certainly. It's really, really interesting. And in fact, things happen in the cytoplasm for the opposite reason. For example, glycolysis. If you know a little bit about glucose metabolism, it starts with glycolysis in the cytoplasm. And that takes the glucose and turns it into pyruvate through a complicated process that involves a lot of isomerases and phosphorylations and all this messy stuff. It's like, why is this so complicated? When you look at that system of glycolysis, it's really complicated. And the reason is because what it's doing is throwing off any kind of deuterium that happens to be sitting on that glucose. It wants to throw it off into the cytoplasm, into the water in the cytoplasm, and replace it with hydrogen. So it's actually scrubbing the deuterium out of the glucose at the same time as it's making the pyruvate. It's very clear. And that's something that Laszlo explained to me, and he's written papers. He's very much aware of this metabolism of glucose and then what happens in the citric acid cycle, which is inside the mitochondria. Yeah. The pyruvate gets delivered to the mitochondria, then it carries forward with the citric acid cycle. And that's producing NADH, and that H is a golden H. It's not going to be deuterium because of all the efforts of all these proteins that know how to avoid deuterium. And then that H is finally given to the mitochondrial intermembrane space through an enzyme called NADH dehydrogenase, which is an enzyme that has its own DNA inside the mitochondria. And it's made there, and then it's used there to put the protons. That H is a proton that's not going to be deuterium because all the different ways that you can get the NADH involve making sure it's not deuterium. It's super, super fascinating.
Yeah, it sounds like the body goes to enormous lengths to make sure that deuterium is precisely where it needs to be and not where it can't be. I wonder, is this the same? I mean, you're talking about glucose metabolism in animals. I wonder if the reverse is same in plants. I presume that, those 36 steps of photosynthesis are, the reason there are so many steps is because it takes exquisite care to make sure the deuterium hydrogen ratio is correct. And there's
I
no
suspect
deuterium.
You're right.
Yeah,
yes.
It seems to me as though, if it's true in us, it would be true in the reverse in plants.
Absolutely. And in fact, what's very interesting to me is the microbes. And I've been looking at the gut microbes, and I am just so fascinated by the gases that are produced by the gut microbes. And people get bloating, a lot of discomfort. They've got hydrogen gas, methane gas, hydrogen sulfide gas. These are all produced by the gut microbes. And people complain about bloating because their gases are not getting, what's happening is the gases are not getting turned back into organic matter as they should. And the enzymes that do that are affected by glyphosate so that they get stalled. And then you've got the methane gas. So methane, they've got the cows. They're killing all the cows because they produce too much methane gas, and it's a bad greenhouse gas. They would produce a lot less, I believe, if they were not being fed glyphosate, because glyphosate is messing up. There's a series of dehydrogenases that are able to select deuterium, hydrogen over deuterium, but also the very big trick is the gas itself, because when the hydrogen producing, so there are microbes in the gut that can take organic matter that has hydrogen stuck to it and yank those hydrogen atoms off and make hydrogen gas, H2, and then release that hydrogen gas. And that hydrogen gas is going to be really, really reduced in deuterium. I found a wonderful paper from the 1960s.
Wow.
Talking specifically about this. And they showed that the hydrogen gas had only 20% of the deuterium concentration compared to the hydrogen that was left behind.
Wow.
It's a tremendous distillation process that's able to get rid of a good part of the deuterium. Really, really remarkable. So then you make that hydrogen gas, the gut microbes do, and then other microbes take the hydrogen gas as a reducing agent. They grab carbon dioxide and they make methane. They can also make acetate, and both of those are really, really critical nutrients eventually for the host.
Yeah.
And the methane, of course, is still a gas, so you've got to capture it. You can't just leave it as methane or you're going to lose it. It's just going to go away. It's going to pollute the environment. So there's other enzymes that can turn it into methanol and then formaldehyde and then formate. And you can go all the way to carbon dioxide again. So you can go all the way around the cycle, and they do this. Carbon dioxide to methane, to methanol, to formaldehyde, to formate, to carbon dioxide, the whole loop. And that looks like kind of a futile circle. But what's happening is you're grabbing that hydrogen gas and sticking it onto NAD to make NADH at the same time. You're grabbing, this is a way to get that hydrogen gas back into organic matter, NADH, ready to deliver to the mitochondria as a golden H that's not going to be D.
Yeah.
Really, really fascinating. And those enzymes, dehydrogenases, are suppressed by glyphosate. There's a paper on E. coli looking at enzyme effect of glyphosate on enzymes, and they found lots of enzymes that were suppressed. A dozen different dehydrogenases were listed as being suppressed by glyphosate.
That's fascinating. I know Laszlo is very well. I know he's interested in particular the enzyme fumarate hydratase. But you're mentioning that there are all of these enzymes that are not only affected by glyphosate, but are also critical in selecting particularly hydrogen nuclei rather than deuterium.
Exactly.
Are these enzymes a way that the body is sort of managing the deuterium hydrogen ratio?
Absolutely.
Right.
Yeah, these dehydrogenases are very interesting because they're members of a class called flavoproteins. And I talked about flavoproteins in my book. And they have a very fine deuterium susceptibility motif at the place where they bind FAD. NAD and FAD are both able to grab hydrogens that are going to be gold, pure gold, because they're not going to be deuterium. And these enzymes, these flavoproteins, they have a very fancy mechanism that involves proton tunneling that's able to pass the proton from NADH to FAD and then finally to make water, for example. They can make deuterium depleted water.
Right.
And they can make it, they can turn NAD plus into NADH. They can turn NADH into water. I mean, they can do all kinds of interesting things that are going to provide low deuterium hydrogen to places where it's needed.
Yeah, that's so fascinating. I guess we're in the quantum realm now. And this is something that I regret not asking Gabor, is that, we're talking about, fundamental particles, basically,
yes.
Which takes us to the quantum realm. And I guess our conversation existed within, the standard physical realm. Are there quantum, have you had to think about this in a slightly different way when you're looking into what is happening with hydrogens and deuteriums? Protons and deuterium.
Yeah, I think exactly. So proton tunneling, is really a very special thing that is at the quantum level. And I think that is a crucial thing that these proteins are doing. It was fascinating for me to read. I've been trying to, the literature is hard. It's difficult to read these papers that talk about the chemistry and the physics of all of it. But it was fascinating to read about, I think it was the cytochrome P450 enzyme, which is another class of enzymes that have this sophisticated ability to select hydrogen over deuterium. And they actually have a hydrophobic pocket that sort of keeps water out, except that it allows a handful of water molecules in, like 12. It's like a number, a very small number of water molecules can come into that hydrophobic pocket, and they line up and form a water wire. And then the hydrogen that's released from the substrate has to work its way through that water wire to get to the product. It's really fascinating. So you can picture the water molecules, and they're being handed another hydrogen from the left. Now they've got 3. They're supposed to have 2. They have to get rid of one. So they get rid of another one that they had before.
So it's like a semiconductor.
Yeah, they're basically grab one, throw one, grab one, throw one. And what you end up with is not the same one that came into the system, right? Some different one comes out. But if a water molecule happens to have a deuterium and it has a hydrogen and it needs to throw one, it's going to prefer to throw the hydrogen because it's much lighter. Just think about having a heavy football versus a light one, you'd rather throw the light one. So it leaves behind the deuterium. And even if the one that came in from the beginning was deuterium, it'll get stuck. It won't make it through that wire. At some point, somebody's going to let it go, and then you're going to end up with hydrogen on the other side. So it's a really cool way to get rid of deuterium, is to pass it through a water wire like that, which is just amazing physics. It's so fascinating.
Yeah, so we're kind of in this zone in history now where not only are we eating and drinking more deuterium than ever before in our evolutionary past, but we're also simultaneously poisoning our ability to regulate this deuterium hydrogen ratio in our body, which sets us up. I found it very interesting in Gabor's book when he talks about, treating cancer patients and then them saying, my blood sugars have gone low after drinking, not even changing their diets, just drinking water that's lower in deuterium.
That's really interesting. And that makes sense because it's healing the mitochondria.
Yeah, so I mean, clearly it's having much larger effects than just regulating cell division and just playing a role in that. It's doing a lot of other things. And it seems as though, wherever there's hydrogen everywhere, a deuterium substituting where it shouldn't be is going to cause issues in the capacity for enzymes to do their work, in all sorts of different things, because it's, the bond strength is different, the weight is different,
Yes.
all of the chemistry is, sort of offset. It's kind of like a whole different atom by itself.
Exactly. Yeah. And it's just randomly distributed, and you need to try to control it. And it's very hard to do that. But one thing that the metabolism has learned to do is to make it as little as possible. In the molecules that are going to be actively passed around and used in metabolism, get the deuterium out as much as possible. And that's what the body tries to do.
So how does the body, let's say we drink a bunch of coconut water, which is high in deuterium. How is the body eliminating excess deuterium? Like how is the body, getting rid of it? Does it come out in the feces? I asked Gabor, how deuterium comes out of the body, because clearly it's coming in and some of it's coming out. And he told me it generally is selected in the amine group. So it comes out in the urine through amine groups in urea. But
that's interesting. I didn't know that. I have not been able to find any data on deuterium levels in the urine, frustratingly, but I have seen that the breast milk is low in deuterium, which is interesting. So to feed the baby, you want to have low deuterium. So it's a good low deuterium food.
That's fascinating.
And saliva is high in deuterium. So the salivary glands are able to increase the amount of deuterium they dump out into the mouth, sort of hoping to get it out of the system, right? So saliva is high in deuterium. And then the blood is somewhere in the middle between those two, between the breast milk and the saliva. But I haven't been able to find anything on the urine. I think I must have something on the urine levels of deuterium. I haven't been able to find it yet, but I should look again.
I asked him, he just mentioned that the amine group tends to have more deuterium in it. So he thinks that that's a way the body is trying to get rid of it, which makes sense.
In the
to
urea,
me.
It would make sense. It would make sense that it would be in the urea. Yeah.
Yeah.
The theory that I have that's interesting is about the gelled water, because I believe the gelled water is trapping deuterium. And not Laszlo Boros, but Gerald Pollack. Gerald Pollack is another person that I've connected with a long time ago, and he's a friend of mine. And he's really, I don't know if you know Gerald Pollack. He'd be a good one for you to have on, because he's done a lot of work with gelled water, and he's done experimental work with gelled water, and he has shown experimentally that gelled water makes a battery. It pushes protons out and makes a battery at the boundary between the gelled water and the liquid water. And I find that really, really interesting. And I would expect that those protons would be deuterium depleted, possibly substantially deuterium depleted, because of the fact that deuterium is heavier. It's just like it's less likely to go out into the hydrogen gas compared to the liquid. Then it's more likely to be in the gel than it is in the fluid water. For the same reason, the gel sort of binds the deuterium more tightly, and it's the protons that become more mobile, and those are pushed out. So I'm guessing that the gelled water that lines all the blood vessels is trapping deuterium and leaving the fluid blood lower in deuterium than it would otherwise be. It's a way to reduce the level of deuterium in the water. That's where the hydrogens are going to be that can react. So that's also an interesting possibility.
Yeah, well, I didn't even have to ask Gabor about whether he knew about Jerry's work, because he brought it up to me. He had just seen Jerry present at a conference, and he seemed totally convinced that what he's talking about is happening. And as far as I'm aware, they haven't worked together yet, but I know there are quite a few people out there asking them to work together. And I told him he needs to send some DDW over to Jerry so he can test whether it affects the EZ capacity of the water. I think most people suspect that it's going to increase the EZ. So we'll just have to wait and see. But I mean, to me, it seems like a perfect synergy of their work. I think by putting their works together, we can understand a lot more.
Yeah, I've tried to get Jerry interested in deuterium as well.
Oh, right. I know he knows about it.
Yeah. So I wish he would take a look at that just to see if those protons that are coming out are less likely to be deuterons. I would love to see that it's actually pushing out deuterium depleted protons, because that's what happens from the glacier too. So I have the evidence from liquid to gas and the evidence from ice to liquid, because when you drink water that has evaporated from the glacier, that's going to be low deuterium water. The deuterium stays with the solid form. So that's the same system, solid, liquid, gas. The only question is gel. I would think it would be the same, that the gel would also trap the deuterium. Just makes sense.
Something I really wanted to ask you, because you're quite a big thinker, and I have a feeling you'd be thinking about this as well. I became fascinated with the fact that deuterium concentration differs based on latitude and altitude. So
Yes,
the closer you are to the equator, the more deuterium there is in the surface water. And obviously the further away to the poles you get, the less deuterium there is. And the higher from sea level you get, the less deuterium there is. So clearly, I mean, you're closer to the equator than myself. So you're drinking water, the foods you're eating are going to be slightly higher in deuterium than further away from the equator. Do you think, and this is my idea, that there's something to do with the environment that is helping buffer the excess deuterium that you're getting on the equator? My thought immediately went to UV light, because there's more UV closer to the equator. Perhaps that's doing something to manage the extra deuterium that is coming in.
That's an interesting thought. I mean, I certainly can think of an easy answer, because, light in general, of course, there's a lot more light. Light in general actually stimulates the growth of structured water in your body. It was infrared light, in fact, that was shown by Gerald Pollack in a study, that infrared light in particular could expand this exclusion zone water, which is the gelled water, by a factor of 4. Really dramatic increase in the gel, in the size of the gel. And so that is also then, if it gets bigger, it's going to be able to better trap the deuterium because there's more of it. So it could be that you get more light at the equator along with more deuterium, but they kind of cancel out because the light is able to make the gelled water hold the deuterium better. I mean, that's just a crazy idea that I have right now. So it's
I was thinking
half-baked.
I was thinking the light is also going to improve the mitochondrial function as well, which is going to produce more deuterium depleted water. So, it seems to me
the other thing is that I think light triggers sulphate synthesis in the skin. I've written about that. This is another theory that I have.
Yeah.
That it triggers, it catalyzes the synthesis of sulphate, and sulphate makes the gelled water. And I believe that autism is associated with a severe deficiency in heparan sulfate, which is the sulfated sugar that actually helps to gel the water. And so the light, I've always advocated sunlight exposure as a treatment for autism, but also as a general health therapy. Sunlight exposure, because the light can help to make the sulfate, which can help to gel the water, which can trap the deuterium and therefore improve the deuterium problem.
Yeah, the sulfation, I mean, I found it very interesting when I first got into your work that you were talking about diabetes as a condition of cholesterol sulfate deficiency. So there's, the idea of it being sulfated, and I think everyone's kind of aware that UV light is very important for creating sulfate on the skin. So, which is why cholesterol levels tend to be lower in the summer when there's more UV around, and higher during the winter. So there seems to be this critical interplay between these things. I always found that fascinating that you were talking about this story when no one else talks about cholesterol sulfate.
Yes, I was sort of one of the big promoters of cholesterol sulfate even before I was looking into glyphosate. That came before glyphosate. I had already identified sulfate as a problem with the autistic kids before I was aware of glyphosate. And then it fit very well because glyphosate really disrupts the sulfur system. And I talk about that a lot in my book, but some of those enzymes that are disrupted by glyphosate, that have that glyphosate susceptibility motif, many of the enzymes involved in sulfation pathways are affected in that way, have a very clear glyphosate susceptibility motif.
Yeah, there's nothing that doesn't seem to affect.
Yeah, I know.
So, I mean, we just spoke about Jerry's work. I'm interested to know how that has impacted the way you deal with water. I mean, is there anything in particular you do with your drinking water? There's obviously a lot of debate about this whole easy thing.
I know, yeah.
I've seen all the footage of the the Naphion and the microspheres. It seems very obvious to me that there is an exclusion zone. Whether he's got the chemistry right, whether he's got the structure of the hexagonal, things right. There is definitely an exclusion zone. I don't think that can be argued. You can argue the details perhaps, but has that impacted the way in which you deal with the water that you drink? Do you use any vortexing devices?
Well, this water is deuterium depleted.
Right.
So I've been drinking deuterium depleted water every day for quite some time now.
Interesting. Have you noticed the difference since having it?
I don't know. I'm generally pretty healthy, so I might feel like I have a little more energy, but nothing dramatic. But even so, I mean, I just do a small amount too, because I just do just this one glass, which is only 1/3. It's because I buy light water that's only 10, and then I mix it with twice as much regular water.
Yeah.
So it's giving me something like 100 parts per million. So it's a very small, treatment. It's a very tiny treatment that I'm doing just as a therapeutic maintenance kind of thing. So, I can't say that I've been healthy, so I guess that's good. As you get older, you always worry about what's going to be coming around the bend. So,
yeah, something that seemed interesting to me when I was reading Gabor's work was that you needed to actually consume low deuterium food and water. And I thought, I wonder why can't you just fast? So it actually just seems that you have to take in food and water that has a low DH ratio so that your body can start to get rid of deuterium and start replacing it with hydrogen. So you actually need to take in foods and drinks that have low DH ratios rather than just not eating at all.
Really good point with respect to the gases and the effect of the hydrogen gas by the gut microbes, and the fact that glyphosate disrupts the enzymes that bring that gas back into organic matter. That's why you get excess methane gas in the cows, which is causing an issue of global climate change, because methane is much worse it's like 30 times as bad as carbon dioxide as a greenhouse gas. And the cows are releasing lots of methane, in part because they're not able to convert it back into organic matter that can be kept in the body. And that's because of those dehydrogenases being suppressed by glyphosate, I think. And then, of course, you get the bloating and the discomfort as well. And you see that in the autistic kids. So I think eating a certified organic diet is going to help to heal your enzymes that are critical for the process of producing deuterium-depleted nutrients that can fuel the mitochondria. So I think I like the idea of eating low deuterium foods, and good choices are things like butter and lard and beef tallow, these animal-based fats. It's quite interesting that those are low in deuterium, and that's actually because they have a pretty direct path from acetate to fat, and acetate is the smallest fat. You have the fatty acids, acetate, butyrate. Acetate, propionate, and butyrate have our 1, 2, 3, and 4 carbon fats. And those are the short chain fatty acids that are produced by the gut microbes. And those fatty acids have a very short path back to methane. So they're going to have very low deuterium because the methane came from the hydrogen gas, which was produced by the gut microbes. That whole process is super important for producing healthy nutrients for the colon. The colon loves butyrate, those colonocytes. That's their favorite food, butyrate, and it's produced by the gut microbes. And glyphosate has been shown experimentally to reduce the amount of acetate in the gut. And acetate then becomes butyrate by the gut microbes. And butyrate depends upon an acid pH. The microbes that make the butyrate are acid-loving microbes. And glyphosate increases the pH of the gut. This has been shown experimentally. Increases the pH, which is going to knock down the ability to make butyrate, which is going to starve the colonocytes. And they're going to have to eat something else, which is going to be higher in deuterium. They're going to have more trouble with their mitochondria. You're going to get colon cancer. All of this can happen. You have this inflammatory bowel. These conditions are all going up exactly in step with the rise in glyphosate usage.
Yeah, that's fascinating. I mean, the gut issues are such a big problem, and it's kind of become normal now to have digestive issues, to be uncomfortable after eating. Have you? I mean, I know there are people out there who use resistant starch as a supplement to increase butyrate in the gut to help manage, food sensitivities and things like that. Seems to be very, very important and butyrate gets most of the cheer, but acetate as well is very, very important.
Yes.
What do you think on having sulfur containing supplements, things like N-acetylcysteine, taurine, methylsulfonomethane? Is there a risk of taking these and not being able to manage the sulfur well? I know this is something that Greg Nye talks about. Is there a risk of taking these sulfur containing supplements without, perhaps the support that's required to metabolize them properly?
Yes, well, that's the problem with sulphite, because sulphite is extremely reactive, and sulphite can get converted to sulphate by an enzyme that is probably disrupted by glyphosate. Sulphate gets converted to PAPS, which is a universal sulphate donor, if you will. And that involves an enzyme that binds 2 molecules of ATP at places where they have highly conserved glycine. So those are going to be vulnerable to glyphosate. And in fact, sulfotransferases, they take the sulfate off of PAPS and they put it onto something else to make, for example, cholesterol sulfate. And autistic kids, there's a recent study that showed that autistic kids had severe deficiencies in sulfotransferases. That's the enzymes that put sulfate on things. And that's why they end up with deficient heparin sulfate. They showed in the brain, these same autistic kids, they had more post-mortem analysis of the brain of the pituitary gland and showed severely low heparin sulfate in the brain of the autistic person. I'm not sure that they were all children who had died. So, and then they also showed the heparin sulfotransferase was deficient in these autistic people. So they showed both phenol sulfotransferase in the gut, phenol sulfotransferase in the blood, and heparin sulfotransferase in the brain were all severely underperforming in these autistic kids compared to the controls. Really striking. And I think glyphosate is causing that. Glyphosate is messing up sulfotransferases. They have a very beautiful glyphosate susceptibility motif. So I can find enzymes that are associated with diseases that then I can look at them and say, yes, this is an enzyme that would be expected to be affected by glyphosate. And in many cases, I can find studies that show that it is. So I'm really connecting a lot of dots when I say these things. I think it's quite interesting to look at the chemistry and to look at the autistic kids and to sort of figure everything out from different directions and looking at the actual protein and the sequence and to see that that's going to be susceptible to glyphosate.
Yeah, it's such a. I mean, you can tell your background in computer science and AI, because that's exactly what it feels like you're doing. You're this analytical approach to things,
yes,
and it seems to be paying dividends as well with all these discoveries that you're coming across.
yes.
I wanted to just back up. I mean, I mentioned a few supplements there before.
Yes, you did. And actually, I think those are probably healthy supplements to take.
Right.
If you don't have the sulfur sensitivity problem, sulfate not getting oxidized, and also sulfite not getting oxidized to sulfate, and also sulfite not getting reduced to methionine. The enzymes, in fact, that was shown in E. coli, the enzymes that convert sulfite, which is inorganic sulfur, into methionine, which is organic sulfur, suppressed by glyphosate in E. coli.
Fascinating. Yeah, I mean, this sulfur thing is not really touched on very much, but it's a good reason for me to eat a bunch of egg yolks every day. So this supplement, it got me thinking that supplements don't vet the amount of deuterium or where the hydrogens are coming from in the substrates of the supplements. There's all these things out there. Makes me think of things like NMN. Which is getting a lot bigger at the moment. If that carries more deuterium residue than would normally come, through the natural way of producing NAD in the body, is there a possibility that we're getting NAD in the mitochondria that has deuterium in it when it shouldn't?
Right, I know that is a big worry, and I'm interested, for example, in melatonin. A lot of people are taking melatonin. Melatonin is very interesting because it's derived from tryptophan.
Yeah,
and it actually is tryptophan packed with an extra methyl and an extra acetyl attached to the tryptophan. Actually, the serotonin, sorry. Serotonin comes from tryptophan, and then the serotonin gets an extra methyl and an extra acetyl attached to it to make the melatonin. And that methyl and that acetyl are both going to be very low in deuterium because of the way the body hangs on to those and passes those around. So melatonin has these two wonderful things attached to it, methyl and the acetyl, that then get metabolized in the mitochondria to deliver the protons. And so if you take melatonin, I've been trying to figure out, I start digging around the patents to see how are they able to make melatonin in the chemistry lab, and is that what they're selling? Because that'll be cheaper if they don't have to harvest it from some biological organism, right? When they sell melatonin,
it's all synthetic.
That's what I was thinking, and that means it's probably not going to have those benefits. It's going to just have the normal amount of deuterium that anything has, which means that that's also just deceiving the body, because when the body sees melatonin, it's thinking in a way that, oh yeah, this is great stuff because this came from this process. We know it's going to be healthy, but it's not true.
That is
so I think you have to worry about that with all of the nutrients that we're taking as supplements.
Yeah, because most of them are synthetic. Even amino acids, they're not naturally derived. So there's no
glycine is another one, by the way.
Process.
Glycine, the glycine cleavage system. Glycine is a good source of methyls. It can produce the methylene tetrahydrofolate, which has that, and the methylene gets converted to methyl tetrahydrofolate, and that methyl gets guarded with its life, right? It's passed all around, put onto the DNA. The whole methylation pathways. To me, that's a process of preserving those beautiful methyls. You trace them back to low deuterium from the way that those molecules were made. The methyl comes directly from methane, and then it becomes methane thiol, and then that attaches to an organic matter to make methionine. And now you've got that methyl that's beautiful because it has a short path back to the hydrogen gas, beautifully low in deuterium, and then it hangs onto it and passes it around with the tetrahydrofolate, and then dumps it into the DNA and attaches it to proteins. I mean, it's kind of thrown around all over the body, right? All these methyl groups.
Yeah.
And it's really fascinating with the DNA. And I was suspecting this, and I finally found papers that show it, that the DNA methylation, do you know how the methyls are removed from the DNA?
No, I don't.
It's hard to find the literature, but I found a great paper. The paper was talking about the fact that these enzymes that do it have a strong deuterium kinetic isotope effect, which means they know how to choose hydrogen over deuterium.
Right.
On top of that, those methyls are already low in deuterium. And what they do is they take the hydrogens off of the methyl one by one. They basically go through that same cycle that I told you about with the methane, the methanol, the formaldehyde, the formate, the carbon dioxide. They do all of that while it's still attached to the DNA. It's really remarkable.
Wow.
And what they produce is succinate. They produce succinate from alpha-ketoglutarate, which is amazing because succinate is able to deliver 2 high-quality protons to the mitochondria. It's a critical enzyme in both the citric acid cycle and the oxidative phosphorylation. It's the only enzyme that participates in both of those parts of the mitochondrial role. And it delivers those 2 protons into the intermembrane space, which are going to be really low in deuterium because they came from this process with these methyl groups that were attached to the DNA. So I think that DNA methylation, part of what it is a storage form of high quality methyls that have low deuterium.
That's fascinating. I hadn't really thought about that, but there are a lot of people out there who use supplements for methyl support, and as far as I'm aware, they are synthetically made, and these checks and balances are not existing in those. I use methyl support as well, so it's got me thinking. I wonder how that affects this methylation pathway if the checks and balances for deuterium are not present. It's fascinating.
Yeah, it was something that I initially, very quickly started to think about the supplements and the difference between natural and synthetic is going to be huge if there is, in fact, this notion that the natural is going to be very low in deuterium. And I think that's true for the methyls, definitely. Especially, and also the acetyls. Both of them are passed around,
Yeah.
And that's another thing that I was fascinated by for the longest time. Why does the body want to stick all these methyls everywhere? It's really interesting. They have an effect, of course. They have a tremendous effect on the DNA. And so highly methylated DNA is protective from cancer. And when the DNA gets hypomethylated, that's when you have a risk of developing cancer. But then in cancer, it's able to actually selectively methylate critical controllers to influence certain proteins to be expressed that are oncogenic that can start to cause the cancer. So there's a whole complicated process. It's amazing intelligence in the biology. It's just mind-boggling how much intelligence there is there, as far as how it knows what to do when. And I think that cancer cells are able to help the body improve the body's deuterium situation in a way by sacrificing themselves. Cancer cell, I've been studying a lot about the cancer with respect to deuterium, and I'm really making headway on that. It's quite fascinating. For example, the cancer cells, they have mitochondria that might even be working fine, but they don't use them to make ATP. They rely on huge amounts of glucose, and then they convert the glucose into lactate. And when they do that, they take an H off of NADH and they stick it onto lactate. And then they send the lactate out to the world because they just throw it into the circulation. The rest of the body, the brain can take up that lactate and it can say, thank you very much for that beautiful proton that you delivered to me that came from NADH that I know it's going to be depleted in deuterium. So it's actually providing the body with deuterium depleted fuel. The cancer cell is, and then it doesn't use its mitochondria, so it can afford to have the mitochondria be sick because it's not really using them to make ATP. It doesn't depend as much on having low deuterium in the mitochondria because it's not using them for that purpose, which makes it less susceptible to deuterium damage.
So it's more like a protective mechanism, which is what kind of what we see in a lot of diseases, is that the protective mechanism is good for a short period of time, but not for a long period of time.
It can't last forever. It's sort of pitching in to try to help salvage the situation. But at some point, it's just things are so bad that nobody can fix it. Yep. And, I think when you treat cancer, when you remove a cancer, now you've lost the protection that the cancer was providing. And then you get things like a bad immune system. Your immune system gets harmed. The immune cells are actually going in to the cancer. And they're not, if there's a problem with the cancer, the immune cells are not actually removing the cancer like they should. That's because they're sick. They have too much deuterium. But the cancer is helping them to fix themselves, just like the virus is helping them to fix themselves in the lungs. And eventually, hopefully those immune cells can actually get repaired by virtue of hanging out in the cancer environment and benefiting from what the cancer cells are giving them. And if they can get repaired, then they can go ahead and take away the cancer. So it's kind of a race against time to try to get that immune system in shape, good enough shape to be able to clear the cancer. And they won't do it unless the immune cells are healthy, and they won't be healthy unless they get rid of their deuterium, which the cancer cell is helping them to do. So it's really, really interesting. But it can only go so far. If you've got so many poisons, you just can't get to where you need to be. Yeah,
that connects really, really well with, why the deuterium depleted water seems to be so effective for cancer. I mean, that story meshes perfectly with what you've just spoken about. So yeah, I mean, the more you think about it, the more the body is just trying to go into self-preservation mode with the resources that it has.
Exactly.
With all kinds of diseases. But we just view it as the body's made a mistake, which generally is the wrong way of looking at it.
You're right. It really frustrates me with the industry, because what they do is they sort of see a protein that seems to be out of whack. That's like it's doing something that looks like it's bad, and then they'll find a drug that can keep it from doing that. Right? They'll poison the protein so it can't do that, and they somehow think they've succeeded in accomplishing something good, but they haven't, because I think all the reactions that the body has are sort of alternative plans for how to cope with the situation. And it's really the poisons that have messed things up, or the lack of certain vitamins and minerals, all the different disruptions that we have because we're not eating well. The body is doing the best it can to cope with it. And when we knock out certain enzymes, we're hurting the system that's trying to help, I think. And that certainly has to do with statin drugs, killing the ability of the liver to make cholesterol, which I think is a terrible idea.
Yeah. Do you think that these prion diseases, like these tau tangles in neurodegenerative disease, these are protective mechanisms?
Yeah, that's a really interesting one. I've been studying them a lot actually lately, because I want to connect it to deuterium. I really badly want to connect it to deuterium, and I'm trying to figure out how to make that work. And I'm suspecting perhaps that there's all these protein misfolding diseases, these interesting proteins that fold in a certain way, and they do perform very important things for the body. But then they can trigger this kind of misfolding. They go from alpha helix into beta sheet form. For example, the prion protein, right? It forms these beta sheets. in totally different structure that kind of locks up into this tight hydrophobic, pile that possibly, I think, traps deuterium. This is what I'm working on as a possible theory, that when there's too much deuterium in the cell, it triggers these proteins to misfold in this way, but at the same time to sort of suck deuterium out of the cell. That's a hypothesis that I'm working on. It could be completely bogus, but I'm trying to make that work. So, because I do believe that I'm kind of believing that everything has to do with deuterium at this point. I just see it so clearly. Even the cholesterol, metabolizing all the hormones. When you look at the hormone system, I mentioned the tryptophan going to serotonin, going to melatonin. There's also dopamine. These all come out of the shikimate pathway, which is the pathway that glyphosate famously disrupts. So you have trouble with the supply of these essential aromatic amino acids that are precursors to the neurotransmitters. But the neurotransmitters, I've been looking at all the enzymes that modify them. Really fascinating, because these enzymes are typically enzymes that are able to select deuterium, hydrogen over deuterium, and enzymes that extract hydrogen from the molecule and make water. That water is going to be deuterium depleted. And these enzymes work in the mitochondria. So everything is looking right to say that the hormone system, these neurotransmitters, not hormones, neurotransmitters of dopamine, serotonin, melatonin, what's the other one? I'm losing adrenaline, right? It ultimately becomes adrenaline. Dopamine turns into adrenaline. And all of those steps are delivering deuterium depleted water to the mitochondria.
Yeah,
that transform those into those other things. So it's really, really interesting.
One of the reasons I got so interested in this deuterium story was because it seems about as foundational as you can get. The ratio of deuterium to hydrogen, you can't get much more foundational to that in biochemistry. I mean, it's there. It's always been there. It's been there for way longer than we have. So it seems to affect all higher order beings. It doesn't seem to affect single-celled prokaryotes.
It's interesting, isn't it, that the microbes can survive on high deuterium? I'm so fascinated by that. Which also makes me wonder if the microbes are able to kind of extract the deuterium out of the body. I do think the viruses can. In fact, I read papers that showed that viruses actually bind the RNA, and the RNA viruses binds deuterium. Really interesting. So they could be trapping deuterium too to help to support the body's need to get rid of it.
That seems like a decent segue. I don't know if you'll be able to give great answers, because I don't think we have great answers for these questions, but you got very interested in, during COVID. I mean, it's good we can sort of look back on that last few years with the benefit of retrospect now. What is a virus? It seems, I'll ask this first. Do we understand viral illness?
I don't think so.
Yeah, I don't think so either.
Yeah. No, definitely not. And of course, also even just the vaccines. And I was aware for long before COVID that I was aware of this, that when kids get measles and mumps, as children, they have long-term benefit in terms of things like diabetes as adults. It's quite remarkable. Just by virtue of having had measles as a child, it's giving you a decreased risk of diabetes as an adult. That is like really, really wild, right? There's something that was done with that infection, I think, that greatly improved the health of your immune system. So I think the viruses are necessary.
I know that things like having natural chicken pox protects from certain cancers quite robustly as well. It seems like these childhood illnesses are a rite of passage for the immune system, in a sense. It is training and curating the functionality of the immune system, and when we prevent that from happening, we don't know what the consequences of that are going to be. I mean, we're in at a time when, autoimmune diseases are higher than ever.
Yes.
Food allergies, all those kind of allergic things where the immune system is not functioning the way that it used to. Even 50 years ago, something's changed quite a lot.
Absolutely. Yes.
Recently. And I know you're quite passionate about this.
Yes.
Particularly with respect to the connection with autism.
Yes.
What have we got wrong with this viral illness story, do you think?
Yeah, I think everything. I mean, I've definitely reached the conclusion. It took me some time. I have to say, my kids were vaccinated on schedule for the most part. I got suspicious of the vaccines just when my youngest child was sort of finishing up his program, and
Yeah.
I got him to avoid his last shot. So, but
Right.
I wasn't waking up until after my kids were grown. Luckily, my kids were grown before everything, got bad, because it was before the 1986 law that was passed that allowed the companies to get a get out of jail free card, basically, that if their vaccine kills the child, it's no problem to them, which is really, really frustrating. And after that, all these vaccines were rolled out. Today's kids are getting completely overloaded with the vaccine agenda. It just breaks my heart. And I think it's really ruining their immune system and accelerating the aging of the immune system. And I got very interested in the thymus, and the thymus in the child is very important at the earliest. The thymus is this gland that is at its biggest when you're born, basically, and it does a lot of work in the first year or two of life, learning about all the different, which cells are your own cells and which ones are foreign, and developing which proteins, looking at the proteins to see which ones are natural human proteins, which ones are not, to learn not to attack the human proteins, but to make sure to watch out for these foreign proteins, and gets all this training in the thymus. And I did actually a deep dive into the immune system in the process of writing a paper about the COVID vaccine, which I find very, very dangerous. And we found some really fascinating things about. We've got a paper that's we're still struggling to get it published, but it's got a very interesting take on the immune system and the thymus in particular, and what happens with the shots, because the COVID vaccine in the muscle, it's got these nanoparticles, and the muscle cells take them up and start making this spike protein, and they release exosomes that contain, basically, it's almost like a naturally generated transformation of the vaccine into these lipid particles that are produced by the cell that can contain the spike protein and can also contain the spike messenger RNA. So they can be distributed throughout the body from the muscle cells. And then especially the spleen is where all the action takes to develop the immune response. And then those dendritic cells, it turns out that some of those dendritic cells that have been transfected with them, because they come into the muscle, they pick up the vaccine, they're also making spike protein, they're in a mess, and they go into the immune system, they go into the lymph system and make their way to the spleen, and they're trying to tell the B cells and the T cells, hey, get some antibodies to this stuff is really bad. But some of those dendritic cells that have been activated by that spike protein actually go back to the thymus. This is so interesting. I read papers on this. They go back to the thymus. And in the thymus, they're now going to start releasing spike protein, because they're still making, it's got that messenger RNA, they're still making spike protein. And that spike protein is going to affect the thymus epithelial cells, which have the ACE2 receptor. They're going to pick it up. They're going to pick up the spike protein. It's going to make them sick. And when you make the epithelial cells of the thymus sick, you get thymic involution, and that is aging of the immune system. So the accelerated involution of the thymus in a child is going to be devastating. So I think that if they start giving the COVID vaccine every year to the school children, then God help us all. I really think it's going to be a very serious mistake. And these kids are going to have accelerated thymic involution, which is directly translated into accelerated aging, period, for the body, accelerated aging. So I think it's going to decrease the lifespan of these kids that are getting exposed to this. If it happens every year, they get another COVID vaccine, it's going to be, I don't want to think about it.
Yeah, that's sort of been my approach too. I haven't looked into this very much at all, because it's quite overwhelming to sort of try and take in and understand. Are there things that you think people could do to prevent any of the untoward effects of these shots?
I know. Yeah. I have friends, who are naturopaths and who are trying to figure out how to treat long COVID, which is really the same thing as vaccine injury syndrome, I would say. Yeah. And, and there's all these kind of antioxidants that you can take, interesting phytoproteins that are made by the plants. There's a lot of plant-based molecules that are quite fancy antioxidant molecules. I mean, I can think of things like resveratrol and curcumin, these are examples, but then there's a whole bunch of others that these people know about that they recommend. Lutein is one that I've got. One of my friends is very excited about lutein, which is found in egg yolk. And he thinks that it's a really healthy molecule to help fight the COVID problems. And of course, I would say lots of sulfur containing foods, lots of sunlight. These are all the things that I think will keep you healthy. Stay away from glyphosate, eat organic diet. I think and soaking in Epsom salt baths is another one that I like. Soaking up the sulfate through the skin instead of relying on your gut microbes to do it for you, because they get messed up by the glyphosate. So, kind of buffering up your sulfur system, because I think that's really important too for the lysosomes. The lysosomes need the heparin sulfate to be able to break down cellular debris. And so part of that is to be able to break down the viruses. You've got to have working lysosomes. And the lysosomes are getting hurt by the glyphosate and the other chemicals that we're exposed to as well. So you sort of have to just eat a very healthy diet, all natural, whole foods, organic, home cooking, lots of fermented foods, lots of sulfur-containing foods. I don't like a vegan diet. I think you should be eating animal foods, especially animal-based fats like butter and lard. So that's kind of my program, is just living healthy. Of course, getting some exercise, getting outdoors, walking the beach if you happen to live near the beach. So that's kind of my formula for health, and it's worked well for me. So
the foundations. Yeah. In the email you sent me briefly after we teed this up, you mentioned something about aldehyde dehydrogenase, and you linked an article that talks about something commonly known as the Asian flush.
Yes, right.
When you
That's
get a
so
flushing of the skin.
Yes.
Can you give me a bit more information about what is possibly going on here? I think you linked it with deuterium dynamics as well.
Yeah, that's right, because that's one of those dehydrogenases that are suppressed by glyphosate. And it's a very important enzyme in the liver for detoxifying alcohol. So ethanol is actually a really good fuel, especially if it comes from the microbes. Ethanol is going to be low in deuterium. And you metabolize it to acetate, acetyl coenzyme A, which goes straight into the mitochondria. So it's like sugar. It's a really good food in that respect. But the trouble is that ethanol gets turned into acetaldehyde by a dehydrogenase. And then acetaldehyde gets turned into acetate by another dehydrogenase. And those dehydrogenases are going to be susceptible to glyphosate suppression, which means that the acetaldehyde is going to build up. And it's kind of like formaldehyde. It's actually sort of a cousin of formaldehyde, because formaldehyde is the one carbon alcohol that's gone into this aldehyde toxicity state. And the acetaldehyde is the same thing at the 2 carbon level. Ethanol is a 2 carbon alcohol. And methanol is a 1 carbon alcohol. Methanol is really toxic. That's because it becomes formaldehyde. And formaldehyde is like an embalming agent. Glyphosate has been shown to upregulate an enzyme that plays a powerful role in detoxifying formaldehyde. That's because formaldehyde is showing up in high concentration. The gut microbes make it all the time, but then they metabolize it right away to formate, and that keeps it from being dangerous. But if that enzyme, which is a dehydrogenase, is suppressed by glyphosate, then you're in trouble. The formaldehyde sticks around. And then you've got this toxicity problem. And that combines it with glutathione and makes a glutathione formaldehyde molecule that then gets broken down by an enzyme that can do that. So, and that's the one that gets upregulated, enormously upregulated by glyphosate. So it's because the microbes are seeing that formaldehyde. But acetaldehyde is sort of the same thing. It's like a cousin of formaldehyde. It's just at the 2 carbon level, and it comes from ethanol. So if you're drinking alcohol and you've got a weak acetaldehyde dehydrogenase gene or protein in your liver, which might just be because of glyphosate poisoning, then that acetaldehyde is going to attack your liver, and you can get cancer, you can get liver disease, and all of that. So
that's fascinating.
ethanol, so alcohol becomes a healthy food as long as you avoid glyphosate. So it would be good to know.
So people who do get the flush, who have a genetic, who have genes that curtail the effects of these enzymes that detoxify the metabolites. If they also are full of glyphosate, that is further damaging those.
Their problem will be worse.
The problem will be worse, and the flushing will be really bad. And over time, I guess that can lead to some very severe consequences.
Yes, that's right.
That's fascinating.
Mm-hmm.
Yeah. Another thing I wanted to ask that I asked Gabor, but he didn't seem to know. He didn't seem to have very much to say. There's a section in his book, I'm sure you've read, I'm not sure if you remember it, where he talks about this single-celled eukaryotic organism that appears to lose its capacity to respond to light in a circadian manner when in a petri dish of heavy water. This made me very interested because I was wondering if it impacts circadian rhythm to have a DH ratio that is elevated. And he also mentions in the same paragraph that there is a melanin producing bacteria that, when put in heavy water, loses its capacity to make melanin or to make its pigment, which makes me wonder, I get a lot of sunlight and I never burn. And I don't have particularly dark skin, but I can be outside pretty much all day and never burn. Whereas I know people who go out for 5 minutes and they're singed. I wonder how much this deuterium story ties into the ability to produce melanin and the circadian regulation of gene expression.
That's extremely interesting. I love what you're saying, and I can't speak about it because I'm not aware of it. I really want to learn more, but that's really interesting that the deuterium is disrupting the ability to make the melanin, right?
Well, I mean, maybe not in humans, but
but in these lower species.
Yeah. So,
that's very interesting.
We know, for instance, that loss of melanin in the substantia nigra is either a cause or an effect of Parkinson's disease.
Right.
So, I wonder how much this is affecting us at those levels as well. We know deuterium ratio impacts on gene expression, and the vast majority of genes have some sort of circadian rhythmicity. I wonder if that's impacting our ability to respond to the day-night cycle, and consequently,
I bet it is,
impact
and
sleep.
I'll bet the pituitary has, and the pineal gland. I'll bet you it has a lot to do with deuterium management. I need to look into that, but that would be a hunch that I would have. Yeah, I'm suspecting that the spleen plays a critical role in delivering deuterium depleted water to the blood. I'm suspecting. I'm working on that idea as well. The spleen gets very affected by high deuterium. There was a beautiful study from like the 1960s or the 1970s that showed that the spleen was very sensitive to deuterium exposure in rats. I think it was rats.
Wow, that's really
interesting to me.
Yeah, I mean, it stands to reason that all of these organs are very sensitive and very involved in regulating all of these things. And
it makes sense to me that the spleen would be. I mean, the spleen is where you get all those dead red blood cells, are metabolized in the spleen. Hemoglobin is actually a very interesting molecule with respect to deuterium because the whole hemoxygenase, which oxidizes hemoglobin and makes biliverdin, and then biliverdin, bilirubin. Bilirubin is a fantastic antioxidant. And the enzyme that converts biliverdin to bilirubin has a strong deuterium kinetic isotope effect. I always look for that, which means that the bilirubin is going to have really good hydrogens that it then oxidates, just like glutathione. It actually gets oxidized non-enzymatically in response to oxidative stress and soaks up those reactive oxygen species, keeps you safe. The bilirubin does that. And hemoxygenase is upregulated under conditions of oxidative stress. And then it creates these molecules that are able to maintain delivering to the mitochondria the low deuterium protons, and protecting them from the oxidative damage. So it's really, really fascinating. Every single pathway that I look at, all these different parts of the human metabolism, I can say, oh, yes, of course, that's going to help out the deuterium problem. That's why I think it's so, so central. It just makes everything make so much sense.
Yeah, it's fascinating how everything is connected when you look close enough. You can trace everything back to everything else somehow.
It is interesting, isn't it? I love biology. It's a giant, giant puzzle, and I'm still poking away at the edges of it. I feel sometimes like I can still be blindsided. I'll come across a paper that says like, oh my God, I had no idea, Still can be very surprised by a new paper. So it's a lot of fun to be able to just rummage through the web looking for papers to read.
I would be remiss if I didn't ask you about AI. I know this
Uh-huh.
is sort of your background. Um,
That's true.
a little while ago, a friend showed me ChatGTP, and I was kind of blown away when I first saw it because I was like, "Oh, this is kind of scary." What are your thoughts on where AI is headed? Are we looking at a point where it'll be very difficult to discern creative output from
I
AI
know.
generated output?
I suspect it's going to be very disruptive, and it's hard to predict exactly what kinds of troubles we're going to run into with it.
Yeah.
I've actually taken advantage of it. ChatGPT, I've enjoyed chatting with it, and it can really, really screw up. I had a lovely conversation with it about taurine, and it
Oh
was
yeah.
telling me how something takes the carbonate off of taurine. And I'm like, wait a minute, taurine doesn't have a carbonate. And I said, you are wrong. And then it apologized and it corrected itself. So it was really funny. And it'll give you say, well, can you give me a reference? It'll say something that I don't believe. And I'll say, well, can you give me a reference for that? And it'll make up a beautiful reference with authors and a title and a journal. And it's complete gibberish, like there's
Yeah.
nothing. So
Yeah.
it's hard. You can trust it on answering simple questions. And it's useful, I think, even for me to write a paper. I've been trying to see how I can use these language models to assist in paper writing, because I would love to just be, I have a lot of papers in my head that need to be get written. And I'm finding some other resources that are built on the large language models. There's something called, oh, I forgot, consensus
Right.
dot something. If you just type consensus, you'll probably find it. It's a platform that an application that some people have put together that is supposed to be focused on biology and medicine and social sciences. But it's better than ChatGPT in the sense that it's different from ChatGPT. When you ask a question, it produces segments of text from various references, and it gives you the paper. So it says, here's what these guys said, and here's the reference. And it's all correct. So you don't have to worry about it hallucinating, which is a great win. And it gives you these papers. So, for example, I can find some interesting papers on a topic that I'm wanting to learn more about by asking consensus. And then it'll pop up these papers, and I can rummage through them and find one that looks, grab it. And then another one is Scriv, SCRIV.ai. That one requires a little bit of effort, because you can actually upload into the application your own set papers. So I can bring a whole bunch of PDF files, say on the pineal gland, for example, and pile them all into a knowledge space and turn the crank. And it'll basically, you can set up a knowledge domain, and then you can have a chat against that knowledge domain. So it has a very specific knowledge that you've given it to it. And you can ask questions about that. And then it acts like ChatGPT in terms of answering the questions, but then it gives the link to the paper and the page on the paper where it found that information. And that's always correct. So it's like a ChatGPT on steroids, where you can specify exactly the papers you want it to look at. And then when it gives you the answer, it can be very helpful for putting it into your paper that you're trying to write. So these are good resources that I found. And then there's other resources that I use that have to do with the proteins, because there's a great one. Shoot.
There's too many to remember.
Uniprot, UNIPROT, I think. Uniprot. I don't know if that's right. My fingers know, but I go there, and it gives you, it's a large database of proteins, and you can search a particular protein, and it'll show you the exact amino acid sequence in that protein. It'll talk about some genetic mutations in that protein. It'll give you the reaction that it catalyzes. All very useful information for me, especially for looking for glyphosate susceptibility motifs, which is really fun. So, and another one that looks at proteins binding phosphate, which is super important. A really beautiful system that you can throw the amino acid sequence for the protein into the resource and push the button, push go, and it comes back and finds places in the protein that bind phosphate, basically. And that's perfect for me, because those are places where glyphosate is going to do its mischief. And it shows you the sequence. And you can see there's a bunch of glycines there. And you can see that the glycines are at the place where it binds NAD or FAD, So that one's been great for me to find proteins that have glyphosate susceptibility motifs. So I use a lot of the AI in my work. And as a computer scientist, it's sort of natural. And it's really helped me, I think, to put together some of my theories that I've been developing.
Yeah, that's fascinating. I guess it's all going to come down to responsible use and a discerning eye.
I think so. You've got to be very careful. The other problem, of course, is plagiarism, because sometimes the chat bot can come back with a lovely story about something, but it might be exactly taken verbatim from somewhere. It can turn out to be. And then you think this is something ChatGPT created, so I can just throw it into my paper.
Right.
But actually you're plagiarizing someone else. So that's a big worry as well. And you don't know who you're plagiarizing because they didn't tell you, So that gets tricky too.
Yeah. Oh, that's fascinating. I always wondered how you found all these glycine substitution motifs.
It's been great to have these resources available on the web.
Awesome. What are you looking forward to looking into in the future? I mean, it's been about 2 years since we spoke, and you seem to have done well more than 2 years worth of research. So what's sort of next on your list? What are you excited about?
Oh my, I've got many different possible papers in my head that I don't have time to write. And I'm collaborating with these people. It's been great actually but it's hard to pull together a paper actually that's coherent on a particular topic. So I have a number of topics in mind that I'm hoping to churn out papers on. Taurine is one. Just on taurine and its health benefits and why, and then also introducing deuterium. I really want to get a paper out, my first paper out on deuterium. I have not published a peer-reviewed paper on deuterium. I've got so much information in my head. It's kind of overwhelming to know even where to start. And then, of course, there's so little known about deuterium that it's going to be very hard to find reviewers. So, and I think the mainstream is going to want to reject it because there's no interest. I mean, I'm really concerned about whether I'll be able to pull that off. But I do want to write about deuterium. I want to write about taurine. I want to write about cancer and how cancer tries to solve the deuterium problem. Of course, autism is still of interest. And we're working right now on another paper, basically, or a paper linking autism or expressing the concerns about the COVID vaccines for autistic kids or for inducing autism in children. I really want to. We're trying to figure out the science there right now. And that's going to take a while. So. And I mentioned the one on the immune system, which is under review. So I'd love to see that paper published, but it's very hard to get these papers published, as you might imagine.
Yeah, because, I can imagine.
Mainstream doesn't want to hear about it. La, 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 la,
Yeah.
They don't want to know. So that's been a challenge. But yeah, I've got a bunch of things in mind. I might even, I dream that I will write a book about deuterium. I'd love to do that. But I need to have enough time. Time is kind of a problem. So,
yeah, well, it sounds like you've got plenty of ideas to work with. And I look forward to hearing them, particularly after they've been peer-reviewed and published. That would be ideal. I'd rather them not go by the wayside and never get published. So, yeah, I look forward to reading them and keeping in touch with you.
Yeah, great. It's been wonderful talking to you, and thank you for taking the time to talk about deuterium because not everybody wants to hear about it.
Yeah, no, I think
it's not an easy topic.
I think it's important because it's something that can easily be, tossed aside and there's nothing going on there. But there's clearly some very important things going on with this, and we need to take it pretty seriously.
I think so.
Yeah. So just a reminder to anyone who's watching. Your book, Toxic Legacy, still one of my favorites.
Okay.
So looking forward to the next one.
Thank you. Yes, I hope I can pull that together. Yeah. That would be great.
Thank you so much for listening. I really hope you enjoyed this one. If you're interested in reading Stephanie's publications, book, or even watching some of her presentations on YouTube, I've left all the relevant links in the description of this episode. If you'd like to support the podcast, you can subscribe so you can get notified whenever I release a new episode. I would also like to encourage you to leave a 5-star review or give a thumbs up if you like the episode. This is a simple, no-cost way of supporting my work and help me reach more listeners. Please feel free to leave comments on my YouTube channel, as I do try to read through as many as I can. I've also put links to all my social media platforms in the episode notes if you'd like updates about the podcast, information about health, or if you'd just like to reach out to me in general. So thanks again for listening, everyone. Take care.